Histology of testis. Testes are male gonads situated in the scrotum. Coverings. Each testis is surrounded by 3 coverings: tunica vaginalis, tunica albuginea, dense fibrous membrane that surrounds the organ; tunica vasculosa, loose connective tissue with blood vessels that ramify in the inner part of tunica albuginea. Tunica albuginea consists of closely packed collagen fibers with few elastic fibers. The tunica albuginea thickens in the posterior part of the testis to form mediastinum testis. The mediastinum testis is not covered by the visceral layer of tunica vaginalis. Septa arise from the tunica albuginea, which pass through the substance of the testis towards the mediastinum testis and divides it into lobules. There are about 200 to 300 lobules in each testis. Each lobule is conical in shape with apex directed towards the mediastinum testis. Each lobule contains 1 to 3 seminiferous tubules. Thus, each testis contains 200 to 600 seminiferous tubules. Seminiferous tubules are highly convoluted tubules. When stretched, each tubule measures about 70 to 80 centimeters in length with a diameter of about 150 micrometer. Microscopic features of testis. The tunica albuginea covers the entire testis. Just beneath the tunica albuginea is a layer of loose vascular connective tissue called the tunica vasculosa. From the tunica vasculosa, connective tissue septa extend inwards and form the interstitial connective tissue. Thin fibrous septae divide the gland into lobules, and seminiferous tubules can be appreciated in each lobules. Interstitial connective tissue binds the seminiferous tubules. The space between the seminiferous tubules are filled by loose areolar tissue which contains blood vessels, nerves, lymphatics, fibroblasts, macrophages, and mast cells. Interstitial cells of Leydig are present in this connective tissue. These are large, round to polyhedral shaped cells. They possess an eccentric nuclei. Cytoplasm is rich in lipid, takes up light stain, and appears foamy as the lipid dissolves during slide preparation. These cells are the endocrine cells of the testis and are responsible for secreting male sex hormone. Several layers of cells are present in each seminiferous tubule, which varies in size and shape. Most of these cells represent various stages of spermatogenesis. Wall of each tubule is made up of an outer layer of fibrous tissue which contain few myoid cells. These cells probably help in the movement of sperms within the tubules. The seminiferous tubules are lined with a stratified epithelium called the germinal epithelium or the seminiferous epithelium, which rests on the basement membrane of the seminiferous tubules. The germinal epithelium is made up of 2 types of cells: proliferative spermatogenic cells. These cells are a collection of cells that exist in various stages of spermatogenesis. Non-proliferative Sertoli cells. These cells are called the sustentacular cells. These cells nourish the developing sperms. Sertoli cells. These are also known as sustentacular cells. These are the supporting cells found in the seminiferous tubules. They are tall, slender cells which are pyramidal or columnar in shape. Nucleus of the cell is basal, which is irregularly shaped and stains light. There is abundance of mitochondria within these cells. Each Sertoli cell extends from the basal lamina till the luminal part of the tubule. The adjacent Sertoli cells are connected through tight junctions. This divides the wall of the seminiferous tubules into two compartments. Deep abluminal compartment. It contains spermatogonia. Superficial adluminal compartment. It contains other cells of spermatogenesis. Functions of Sertoli cells. It supports the germ cells, provides nutrition to the developing germ cells. They phagocytose the residual cytoplasm during the spermiogenesis. In fetal life, it produces a hormone known as Mullerian inhibitory substance, which suppresses the development of Mullerian duct. In adults, it secretes androgen-binding protein, which binds the testosterone and makes it available to the germ cells in higher concentration. Form blood-testis barrier. Blood-testis barrier. A junction is formed between the Sertoli cells by the gap junctions, tight junctions and adherence junctions. This junction acts as a physical barrier between the seminiferous tubules and the blood vessels, thus isolating the developing germ cells from the underlying blood supply. This barrier divides the seminiferous tubules into two parts or compartments, namely, basal compartment, seen on the outer side of the seminiferous tubules as in contact with blood circulation and lymph. Adluminal or apical compartment is seen at the inner side of the seminiferous tubule. Spermatogenic cells. Primitive spermatogenic cells are arranged towards the basal lamina and the developed cell towards the lumen. In the order of their maturation from basal lamina towards the lumen, spermatogonia, primary spermatocyte, secondary spermatocyte, spermatids, and spermatozoa can be appreciated. Spermatogonia. These are the stem cells from which the spermatogenesis begins. They lie near the basal lamina. It undergoes mitosis and gives rise to numerous spermatogonia. There are two types of spermatogonia. Type A spermatogonia. Based on their staining property, they are distinguished as dark type A cells and light type A cells. These cells have an oval nucleus with eccentrically placed nuclei. Type B spermatogonia have spherical nucleus with spherical nucleolus situated in the center. Each type B cell divides multiple times to give rise to primary spermatocytes. Primary spermatocytes. These are the largest cells. The cells occupy the central part of seminiferous tubules. Each cell consists of large spherical nuclei. The primary spermatocyte undergoes first meiotic division to give rise to two secondary spermatocytes. The first meiotic division helps in reducing the chromosome numbers to half. Secondary spermatocytes. These are smaller than the primary spermatocytes. They have a smaller nuclei in the central part of the cell. It carries haploid number of chromosomes, 22X or 22Y. Each secondary spermatocyte gives rise to two spermatids through second meiotic division. Appreciating secondary spermatocytes in sections is a difficult task as they divide immediately to form spermatids. Spermatids. Spermatids are round cells with spherical nucleus. The cells and their nucleus are smaller than the previous cells. They lie in group in association with the Sertoli cells. Spermatids forms the mature sperm or spermatozoon. Sperm. The process by which spermatids transform morphologically into spermatozoon or mature sperm is known as spermiogenesis. There is no division involved in this process. The sperms can be seen in close association with the apical part of the Sertoli cells. In fact, the head of the sperm appears to be embedded in the cytoplasm of the Sertoli cells. The process of release of sperms from the Sertoli cells into the lumen of seminiferous tubules is known as spermiation.